Hello. There's no ring, but I think it's time. Should I just, yes. Right? Okay. So first, thank you so much for coming here. This is always a difficult time after lunch. I hope that you can bear with me. I'm going to show you today research that is, well, some of it is a little bit old, some of it is new, in a very, very cool system. The questions that we're trying to answer, they come from these observations that people have done in the field for several years, decades actually. Perhaps one of the most striking in this particular system is transparency, right? Glass frogs, you see a glass frog, and the transparency level that some of these species reach can be actually like mind-blowing. If you pay attention to that particular picture, you can see the heart, you can see the liver, you can see the whole digestive tract, even part of the circulatory systems, and even the bones in the fingers and the toes. It's really, really amazing. There's also a lot of variation in terms of parental care, and parental care varies a lot from, in one extreme, fully maternal care, where a couple of species take care for during weeks for their offspring. On the other extreme, there's fully also male parental care. And in between, there are many species where they have very, very short maternal care that is just a couple of hours, and then they flee the scene. So that's a particular trait that also we're going to study. There are very weird things in the system. This is a male of the largest glass frog species that there is about this big. And if you pay attention to the arms, they have these bony structures that come out. So that's another very peculiar trait that is present only in adult males of some species. We have many questions about the place of origin of this neotropical group. We also have questions regarding speciation patterns, and of course, trying to understand things about why some places have more diversity than others. I'm not going to focus a lot on methods. In this part, we only need basically two things. We need good data on one side, and then we need a good tree on the other side. And all the other things are, well, this, of course, calibration and good models. And we can talk about that later on if you want. But basically, we need a good phylogenetic framework in order to be able to answer these questions. And this is perhaps the area where we've had more changes in these last 20 years. I graduated from my PhD in 2008. And the first paper that I published with a phylogenetic tree is on the lower left, and it had 11 morphological traits. So really nice. Now our working tree is the one that I'm showing on the right. It has more or less 6,500 base pairs with a combination of mitochondrial and nuclear genes. And now we are jumping to the genomics. We still don't have those results. I thought that we were going to have them by now, but this is still our working tree. So this is a moving target, right? In terms of genes and in terms of also in the species that we can get to have a representative sampling. Right now with this tree, we have about 70% of the described species, so that's pretty good. One of the highlights, I think, I enjoy taxonomy a lot. With this new tree, we were able to describe 7 new genera of glass frogs, so that was a beauty, and we placed names as we wanted, and nobody could argue about those names. So that was a fun part. Then we started to understand these diversity patterns, right? And what is clear, what is very obvious in the species distributions, is that these things like mountains a lot. And the northern Andes is the biogeographic region that they enjoy the most. Half of the 160 species are found in the northern Andes. So if you ever want to see these creatures in the field, you have to go to either Colombia or Ecuador. Go to Ecuador. It's rare. Yeah. When working on the origin of the group, with no ambiguity whatsoever, we can tell that South America is the origin of this group. And then we've had several independent dispersal towards Central America, at least 11 with the working tree that we have right now. We have been able to also understand a little bit of the past history. So the ancient areas that were glass frog inhabited were these more or less cloudy mountain forest between 1,000 and 2,000 meters of elevation. Even though we were expecting to have a big influence of the Andes, there's no peak in terms of diversification that is associated to the uplift of the Andes. In terms of the origin of the family, genera, and species, we have the family origin in the Eocene, most genera in the Miocene, and species mostly in the Pliocene and Miocene. There are some species that we are describing right now that are a little bit newer, more recent from the Pleistocene. In terms of speciation, as many other groups, it's been shown that there's a lot of niche conservatism. There are many similar species that are clearly independent lineages that are evolving in very similar environments. And when we plot diversification rates and climatic niche evolution, we don't see any trends. So basically that means no significant niche evolution. And even at the generic level, when we place the distributions of many species on the map, you can see, for example, that most of the central species are restricted in the northern Andes, and genera as diverse as Nymphargus, almost 40 species, they also have very restricted distributions. You can even find sister species that are at both sides of the Andes, so in the Chocó ecoregion and the Amazon basin. So really strong niche conservatism. And of course, if you combine all these things, if you combine niche conservatism, if you combine the topographic complexity of the Andes, and taking into account that tropical species tend to inhabit certain climatic bands, at the end you have a scenario that of course is going to promote a lot of speciation, right? You're going to find a scenario if you, in the upper panel, if you see an ancestral species with that distribution in the Andes, starting with some random extinctions and then starting with some erosions, you start having some limits to gene flow. So in the same basic elevational band, you can have a lot of interspecific variation because of geography and because of these limitations. And later on, you can start having a lot of species that are basically very similar in terms of morphology, but that they don't have already any gene flow because of these barriers. And the ending result can be problematic and can be, I was listening to the opening session by David Bone, and of course, yeah, you get, okay, yeah, there are no species. You can get to those conclusions because it can be problematic, even though no one said that taxonomy was going to be an easy task, right? But I think most of us, it's like who likes Coldplay? Nobody said it was easy, right? But we do have these lineages, and those lineages really are cohesive, and they have a history. And in this never-ending species discussion, I think we should name those species. I think that's a big part of our responsibility. We're also thinking about conservation. So we have the tree, right, and we have these traits, and the evolution of these traits is mostly complex. There are no cases where you have a single origin of a particular trait, so transparency, it has evolved multiple times in the tree. There are a couple of very good papers that are explaining transparency. So one of them is by Barnett et al., which basically says that transparency helps to break this outline of the frog, and that helps camouflage for these species, and of course avoids predation. Even a cooler paper by Taboada and Deli and et al., they show that when our glass frog is resting, up to 90% of the red blood cells accumulate in the liver. And of course, that creates tissues that are much more transparent. So this is also a very nice paper if you want to know the mechanisms behind transparency. For morphology, again, this humeral spine has evolved multiple times in the tree. And even though these things are mostly tiny, they're like this big, they are ferocious, right? They have these huge fights for days and days. Anna is here, she's nodding because she's seen this in the field. And they're fighting for reproduction places, these places where females at the end can select for reproductive activity. The perhaps most interesting part of this whole story has to deal with parental care. As I was saying there, like in green, you have clades where you have extended male parental care, and in the other, in black, you have maternal care that can be extended or can be very short. Is not just inference. There's a lot of observations that actually show that this close association between either males or females with the offspring really provide a significant benefit in this case. They avoid mortality by reducing a lot desiccation, or also there's a couple of nice videos where you can see the actual glass frog like kicking out wasps that are trying to eat the eggs. So it's very active. It's really, really meaningful in terms of the survivorship of the offspring. And I think we all understand more or less the origins of maternal care, right? You have this initial investment, and then you are stuck with it. You have to keep investing. So sorry, but now you have to. So here, there's also a big investment by males. So we have to create a scenario of how this paternal care has evolved. And this is really, really cool. So Annie, she's like crazy in the field, and she's measured tons of museum specimens. And what she found is that good fathers have small testes in glass frogs. If you don't believe me, measure yourself, right? See what happens, do the correlation. But this is what we found. Good fathers, they have very good, very short, very small testes. Bad parents have large testes. That makes sense because testosterone, and if you have low levels of testosterone, and of course there's a little bit of speculation, but you have higher degrees of parental care. So in a perfect system for females, females should choose those males that have small testes, right? Because that would mean that that male is going to take care of the offspring. That's the ideal scenario. But how do females make that inference? Okay, they are transparent, but they are not that transparent, right? It's not. It doesn't work. It doesn't work that way. Another incredible study by Annie. She showed that in the field, females would choose for offspring males that already have eggs from other females. So they see a male, they see that male is with egg clutches associated to him, and they prefer that male. So in that way, they know that that male actually takes care of the offspring. So this is a very interesting way of how, most likely, females are driving parental care by males by selecting these males that already have these clutches. So it's a very direct way of measuring good parents versus bad parents. So that summarizes many of the histories that I wanted to tell you about glass frogs. There's always a couple of last slides that I want to say regarding conservation. It's very interesting that now in many countries you have this nice association between endangered species and some kind of protection. And you have that here in Canada, you have that in the US, you have that in many many Latin American countries where legislation protects these species. And with all these species that me and collaborators have described, we use some of them, the ones that are really endangered, to actually protect land and avoid mining activities. So this has become a system where you can actually link in a very nice way a study system, endangered species, and conservation. So I think that is part of our responsibility to just try to make these links, between the organisms that we like, that we love, that we study, and conservation activities. And with that, I want to thank a bunch of people, and many of them are here in the room. And if there's time for questions, I would love to try to answer them. Thanks. Yes.
Yeah, great talk. So small testes has been invisible for us because it sends a signal to the females. But what are the costs associated with being small testes male? Like there has to be some costs.
That's a great question. Perhaps they have a lower ability to actually retain territories because other males can be more aggressive and actually gain those territories in fights. So there can be a cost that is associated to having these low testosterone levels. Yep. Any other questions? Okay. Yep.
When they have maternal care, do the females still have maternal care, or is that lost as well?
So in the species where maternal care is present, so there's, as far as we know, there's no variation within a species of how care is provided. So in some species, it's just females. In some other species, it's just males. Perhaps there's some in between. It would be interesting to see if there's, but we still lack a lot of information about specific behavioral cares in most of species. This is inferred from, I think, it's got to be about 50 species that we have good information. So we still lack 2/3 of the whole diversity of behaviors. Okay, thank you so much.
Hello everyone. I'm Alan Flatres, PhD candidate at the University of Western Ontario, Canada. And today I will present the selective advantages of redirected helping in a viscous population. One question you can just directly ask is, what is redirected helping? So it's a behavior, altruistic behaviors happening in different bird species. So for example, you have the long-tailed tits, so western bluebird or Iberian magpie. And so if you consider a bird individual, it will lay some eggs, but sometimes without, with some bad luck, a predator will come and eat all the eggs. So that's kind of sad for them, but they can decide eventually to help one of the related neighbor to have either higher survival or higher fecundity. So if you know this Hamilton's rule about evolution of altruistic behavior, you have the benefits of helping times the relatedness between the helper and the helped individual. So minus the cost of helping. So if this sum is positive, then all good. Like we have selection of helping. So that could be the end of my presentation if we didn't look at viscous population. So basically when individuals are dispersing relatively close to the natal site. And so in that case, it can create some additional costs and benefits. And as you can imagine, because like individuals will compete with the relatives. And as you can imagine, it may be not the best strategy to help someone you are competing with. So my question here are, how does this population viscosity impact the evolution of related helping? And especially if I'm looking at different spatial structure, this will change how individual will disperse, and it will eventually change the population viscosity we are studying. And finally, I want to look at how the nature of the benefit, so whether I'm increasing the survival or the fecundity of my helped individual. So for example, if I'm helping to feed the offspring, so how these different benefits can affect the evolution of helping, of redirected helping in that case. So to do that, I built two different models. So basically the life cycle is very similar between those two, but I used first an infinite island model on the left side and a stepping stone model on the right side. And so basically in each case, you have some individuals who will face the brood because of a predator, for example. And these individuals can decide to help a rated neighbor. So for example, in the case of an infinite island model, they will help someone on the same patch as a chair. But in the stepping stone model, they will help just a direct neighbor. And so after helping, the offspring will mature, and they will decide to disperse or not. So they will either disperse to another island, or they will remain on the natal island. Or in the case of stepping stones, they will disperse to the closest neighbor, so just one step away. And so we really have this population viscosity as we have individuals which are helping each other, but we also have individuals that are competing with the relatives. And so it may not be good to have to help someone you will be competing with, or at least that your offspring will compete. Just to know if it's good to help someone when you brood fail, we first compute the fitness of an individual, which depends on the neighborhood of this individual. So, for example, if we have different, if the neighbors succeeded in the brood or they failed the brood, this will impact how basically the fitness of the individual and the fitness of its neighbors. So if basically everyone failed on this island or around the focal individual, then it would be some different changes in the different fitness for the individual and for its neighbors. So here we just, for both models, we compute the fitness of the focal individual and weight this fitness by the probabilities that the environment, so the neighborhood, is happening. And so given that we have this fitness function, we can look at the fitness change when for example, the focal individual decides to help someone else. And so this will impact the individual itself, so it will pay a cost basically of survival, but it will also impact the different neighbors impacted and which are related to the focal individual. And so we have this neighbor-motivated fitness change. So we have the sum of the fitness changes for the different neighbors times the relatedness between the focal individual and the individuals impacted by this change of fitness. So basically, if we have this fitness change, we can look at the sign of this fitness change, and if it's positive in a selfish population, sorry. So if it's positive, then we can have a selection of redirected helping, but if it's negative, then we don't have any selection and we just remain selfish. So we can first then look at first results. So when we don't have any blood failure in the population, except on our focal individual, because we need someone to help someone else. And here, I don't directly look at the fitness change. I look at the critical cost-benefit ratio, so associated with different benefits. So survival on the top and fecundity benefits below. And I compare these two models, so this infinite island again on the left side and the stepping stone on the right side. And so the critical cost-benefit ratio is, in a way, like the potential for helping to emerge. So if it's high, then it's easy to have redirected helping. But if it's low, then it's much harder. Like you need a much more benefits, basically. And so if we compare the two different benefits, we first see that so the survival benefits are getting better when we increase the survival of breeders. As you can see, like on the top one, so when we increase the survival, the potential for helping is getting bigger. But when we look at the fecundity benefits, we see that it's decreasing with the survival. And so as basically if we compare these two, as you can imagine, if we have a low survival, it will be better to have fecundity, but fecundity benefits. But if it's high survival, then it's much better to have survival benefits. And so this works for both spatial structures, so the infinite island and stepping stone model. But now we can also look at how the dispersal rate will change, so will affect the evolution of redirected helping. And so if we, in that case, we have differences between the two models. So in the first model on the left side, when we increase the dispersal rate, so going from the black curve to blue curve, when in that case, it's just like we decrease the critical cost-benefit ratio. So it's just basically harder to have emergence of helping. But in the contrary, on the right side, we see that when we increase the dispersal rate, it's better for emergence of helping. So we have differences in how dispersal will affect evolution of redirected helping. So one will be negative on the left side, and on the right side will be like a positive relation between dispersal and evolution of altruism behavior here. So to continue on that, what happens when we increase the brood failures probability? So basically, not only the focal individual, but also other individuals in the population can fail the brood. So that's for them. So again, we can compare how fecundity benefits and survival benefits can promote help or not. And we see that similar results to what we had before. So like for low survival of breeders, it's much easier for fecundity benefits to promote helping rather than survival benefits. But when we increase the survival, so this, and again, we have this S area here, then survival benefits will become better. And we also have an effect of the dispersal rate, again, so similar to what we showed before, that when we increase the dispersal rate, it has different effects between the two models. So in the first model, when we increase the dispersal rate, the range of parameters where we don't have emergence of helping, so this zero area here, so like for brute failures and survival. So in that case, when we increase the dispersal rate, this area is increasing. So that's something we could expect from the previous results, that we have a negative relation between the dispersal and helping, so redirected helping emergence. But on the stepping stone model, so on the right side, we have the opposite relation. So when we increase the dispersal rate, so going from top level to top panels to lower panel, we see that this zero area is decreasing. So again, we have a positive relationship between the dispersal and the emergence of redirected helping. So just to conclude, so I've showed that redirected helping is possible even in viscous population. And it's especially true because of this overlapping generation. So you have endurers that are surviving to each time step, basically. And we also have some specific life history constraints. So like the endurers who wants to help has to fail its brood to help someone else. And in that case, this endure cannot pay any cost of fecundity because it doesn't have any fecundity. And so this change, this life history constraints makes it relevant, make it good to help someone else, even if the survival is low. And secondly, like the difference between our models was about the spatial structure. So is whether either infinite island or a stepping stone. And this change of spatial structure really impacts how the dispersal will affect evolution of redirected helping. And so in a more general, how we model this, the evolution of behavior, of the social behavior, will depend on what kind of spatial structure we use. So especially if we are looking at a competition between individuals, eventually reproduction and other social behaviors. Finally, the life history constraints that we have, so about this cost, and so this cost of survival, for the helper makes that survival benefits can be a better promoter for redirected helping than fecundity benefits, which is something was not shown in previous studies. So, like just basically other altruistic behaviors. And so we really have like how this life history constraint can really change how we can have different results on the evolution of altruistic behaviors. So one possible direction following this project would be to use a different spatial structure again, but here would be more something that will fit real data. So for example, we have an example of long-tailed tits, which actually do this behavior. And so if we have a spatial structure that takes into account that individuals are making flocks before dispersing, then may eventually have, again, different results here. And finally, again, in this long-tailed tits, we also have a load lightning behavior. So in a way, it's quite strange that some individuals are selfish towards their own offspring, but are altruistic towards the offspring of others. So yeah, that could be interesting to see how these two behaviors can evolve. So finally, I want to thank Jeff Wild, my supervisor. I don't know where he is, so hi. And all the people from the Flower Hour Maths Bio group at Western, and so Enter for funding, and all of you for your attention. Thank you. And yeah, I guess we have made time for questions. Yes.
I was just wondering if you let a dispersal evolve, do you expect dispersal to be different between the two models?
Yeah, eventually, because like since we have like like different positive things from dispersal, it may be good to have like in the right side. So stepping stone model, we may have more dispersal than in the left ones, and we have we have less skin competition basically. Yeah. You have a question?
I was wondering if a limit case of your model could bring you back to the classical altruism model and see how it differs between this specific type of altruism.
Yeah, it was something that could have been done after too for as potential direction. Yeah, I guess if we have potentially we could have this, but we really have to modify the life history constraints that we have. So I guess we can have like even a coevolution of regulated helping itself and altruism as its own altruism. But yeah, I'm not sure it could be we could have limit cases from our model. Thank you for attention.
Okay, hello everybody, I'm Nic Bone. I'm a fifth-year PhD candidate in Joseph Huerta's lab at Virginia Tech, and I'm going to talk about some stuff that we've been doing with Elliot Miller, who's now at the American Bird Conservancy. So I'm going to talk about migration today, and I'm going to start out by just kind of going over what I learned as somebody that wasn't really a bird expert in a non-bird lab, learning about migration, which is really that there's a ton of different ways that you can measure the trait. So some people are banding individual birds and looking at how birds are moving throughout a year. Other people are interested in like the orientation or direction of migration and maybe how that changes within a population or how that evolves over time. On a more like macro scale of things, people are looking at radar data and trying to estimate how many birds are moving at any given time, or coming up with these really detailed like species distribution maps that take in all the occurrence data that people know about an individual species. But what I found as somebody that was more on the comparative method sides of things is that once this gets condensed down into like a species level data set, oftentimes people just condense all this information into whether a bird migrates or whether it doesn't migrate or some other version of discretization. And so my question became, where did all this like information go, right? It seems like there's a lot of biology going on the left side that doesn't end up into our character states. So I'm going to do exactly what I just complained about and give you some character states to orient you to migration, specifically from this paper, Ayers et al. 2017, which I think does a really good job describing a lot of this variation. So the majority of birds are actually resident, which means that they have a static range and they're not really doing that much within it. So this is almost, I think it's like 80% of birds are completely resident. Okay, and the next biggest chunk of species are obligate migrants, which are doing the kind of migration that you might be familiar with, where they start in one region, migrate to another region during another period of the year, and then return back. A smaller fraction of species are doing something like partial or eruptive migration, which is considered either a proportion of the population moving during one period of the year or like a full population level eruption where seasonal, when they're tracking some sort of resource that varies seasonally. A really small percentage of birds are considered completely nomadic, which means that they're following some sort of resource almost all the time. They have this completely irregular movement. And my question was sort of, where did all of these different types of migratory behaviors come from? There's been a lot of people that have been interested in this for a long time, but one of the hypotheses that I think is really interesting is that site fidelity, or the likelihood that a bird's going to return to the same site year over year, is a really important driver in the evolution of migratory behavior, which comes out of this paper, A Long Winter for the Red Queen. So site fidelity, like I said, is just describing how likely a bird is to return to the same site year over year. So something with high site fidelity would look like this, where we have occurrences over years, multiple years in the same month, and we're getting the same bird year over year. So we keep seeing that same bird. Meanwhile, something with low site fidelity would have this really irregular pattern of distribution, right? Some years it's there, some years it's not there at all, other years it might be really prevalent. So this made me think about what this might mean on the species level, since site fidelity is often described on an individual bird level, so like within a population. And I came up with this diagram of redescribing those character states that I described before along this site fidelity axis. So at the top we have high site fidelity, and at the bottom we have low site fidelity. We have residents up here with high site fidelity since they're staying in the same spot year round. Then things like obligate migrants also having high site fidelity since they're returning back and forth between the same two sites year over year. Somewhere in the middle, we have a lot of these partial and eruptive migrants, where some of them might have some high site fidelity behaviors and other ones are doing more nomadic movements. And then at the bottom, we have these really low site fidelity nomadic migrants. This made me start to wonder if we could find a measure of this specific axis, right? This like site fidelity axis. And to be able to do this, we would need to gather a couple pieces of data. Specifically, we need to know what species of bird was seen somewhere, when it was seen, where it was seen, and we would need this information for multiple years. Luckily, there's like a bunch of bird nerds collecting this data constantly and putting it on the internet. So this makes this part of the process really easy. So what I did is I took eBird observation data, which includes species, count, latitude, longitude, and date time, as well as a sampling effort data set that eBird provides. And then I split the world into 36 kilometer squared hex grid cells using this H3 package, which looks something like this. The first thing that I did was overlay the sampling effort, which is like the total number of eBird observations recorded at an individual hex onto these hexes. So you could say like there's like 8 total eBird checklists submitted at that hex or 10 in that other hex. The next thing I did was look at an individual species. So here's a scarlet tanager, and then took a specific month and year and plotted out all of their observations at those same hexes. We did that for multiple months and multiple years, which we consider observations. And then we calculated this proportion of observations to sampling effort, which you could think about like a sampling corrected occurrence. So this is per hex grid cell per month per year. The next thing that we did was fit a linear model to predict this occurrence proportion based on a couple different variables. So we had some fixed effects, which were basically monthly variation as a function of latitude and longitude and season. And if you want to talk about that later, we can. As well as random effects of yearly variation and location, so the specific hex grid cells. So you can think about the fixed effects as being the consistent sort of predictable influence on species distributions from seasons, or from months specifically. And then the random effects as being all the other variation that we couldn't explain via that monthly variation, but could be explained by what year it was or what location it was. But let's get back to the question here, because that's not really a metric of site fidelity, that's just a model that we fit for individual species. What we calculated next is this Shannon entropy value, which I think about is like how much information something gives us based on those fixed and random effects. So I calculated a monthly entropy effect, which is basically how much information a specific month gives us about a species distribution. This would be really helpful for things like residents and obligate migrants, where knowing the month gives us some information about what they're doing. Similarly, we calculated a yearly entropy score, which is based on how much information an individual year gives us. So like knowing that it's the 2017, what does that tell us about a species occurrence? And this is really helpful for nomadic migrants, where they have some years where they're really obvious and other years where they're completely absent, and partial and eruptive migrants, which often have these sort of like big eruption years and low eruption years. So what I'm going to show you now is the results of the monthly entropy, which again is whether month explains change in occurrence. So on the x-axis here, we have month, and on the y-axis, we have entropy. Things that have more entropy in this case are like less predictable, so it's more spread out across its range. It's harder to tell where it is. And then things with low entropy are more predictable. I'm going to show you for a tree swallow, which is an obligate migrant. And you can see we have this really nice seasonal pattern where the entropy peaks in the wintering and migratory periods and then goes down during the breeding season, where they're usually returning to the same sites year over year. What I noticed is that you could tell the difference between nomadic migrants, which have this like consistently high entropy score, and things like, nomadics and residents actually look kind of similar, where they just have this consistent high entropy score. But if you compare nomadics to obligate migrants, you get this difference between the monthly variation and the monthly variation not existing in the nomadics. obligate migrants and eruptive migrants actually do look kind of similar, which tells us that there's some seasonality to when eruptives are becoming less obvious in their range, right, when they're doing this eruption behavior. So next I'm going to show you the yearly entropy, again for a tree swallow, which is an obligate migrant. And you might be able to tell that entropy is just increasing pretty much year over year, which I thought was interesting. And then I have found that that's pretty much what's happening for every species that we were looking at. But if you squint really close, you can see that some of them are doing way weirder, upward movements here. So what we wanted to calculate next was some measure of that variation, right? The variation in year-to-year entropy, which we calculated by taking the difference between 1 year and the previous year. So here's the residence year-to-year entropy plot. So the x-axis is years now, and the y-axis is the change in entropy. You can see there's not that much of a change. Versus something like a partial and eruptive migrant, like this, where you have big peaks and sort of troughs depending on what year it is. And versus an obligate migrant, which again looks a little bit more like a resident, where it's not doing that much change year to year. So that begs the question, is this something like site fidelity? Again, just to remind you of our prediction, we have the site fidelity axis. Residents and obligate migrants are kind of doing something similar. Eruptive and partial migrants are in the middle, and we have nomads at the bottom. And that's basically what we see here, right? So this is for about 26 North American species, which isn't a special number. I just ran out of time to fit. And if you look at the obligates and the residents, they both have fairly high site fidelity. They're not changing that much year over year. And then eruptives are sort of this intermediate. And although there's only 2 nomads, and there's arguably only 1 nomadic species in North America, they look like they have way bigger swings in this entropy value year over year. So just to conclude, and some future stuff that we're interested in, this year-to-year entropy value might be a site fidelity metric, especially for the kind of stuff that we're interested in. Specifically, we could use it to try to understand more nuanced phylogenetic patterns, which was the ultimate goal of this metric, than just using those discrete character states that we were looking at before. And that gives us a possibility of looking at things like clade-specific evolvability of site fidelity. So maybe some clades can switch back and forth between things like nomadic movements and less nomadic movements. And maybe that would confer some sort of protection against climate change or something like that. And additionally, we can see if degrees of this site fidelity axis actually have unique life history trade-offs, which isn't necessarily accessible if you're looking at just the character states. And I think, yeah, with that, I'd like to thank you for your attention and thank all these people for helping out. Appreciate it. Yeah.
So I'm curious about trying to sort of scale the individual level site fidelity process, which is like the Red Queen paper.
Yeah.
And this like species level one. How do you see them mapping onto each other? Because they can be driven by totally different environmental properties, I think.
Yeah, definitely. Yeah, I haven't gone far enough to know exactly how we would do that. But there's been people who have brought up like population dynamics and how that would affect the species level site fidelity thing, which I think is a really important point. I'm not sure exactly though, but I'd be interested to find out. Good. All right. Oh, sure.
Why is entropy increasing over time?
I think even though it doesn't necessarily make sense with the using like the sampling corrected occurrences, I do think the fact that there's so many more people using eBird now than there were even 5 years ago might have something to do with it, specifically about like what hexes are being even accessed by species. And the fact that I see that for the residents especially is like kind of indicative of that, because it's like a northern cardinal. It's like how many more houses are reporting a northern cardinal now than they were in 2015 or whatever. All right, thank you.